All cells are produced by cell division. Mitosis and meiosis are two forms of cell division that serve different purposes, but follow similar sequences. Cell division serves numerous purposes. Organisms grow and repair their bodies through cell division, and it is a necessary process in all forms of reproduction. When a cell divides, it goes through a sequence of steps to ensure the new cells have all of the cellular structures necessary in order to function, including, most importantly, DNA. DNA is replicated using a cell cycle phase that occurs prior to cell division called interphase. This phase basically refers to the stage of the cell cycle when the cell is not actively dividing but functioning according to its particular purpose. The division of the nucleus is a process called mitosis, which produces cells that are genetically identical to the original cell. Mitosis occurs through a sequence of steps called phases. The process begins with the cell that divides, which is called a parent cell. This image shows a very simple summary of the process of mitosis and cytokinesis, where one cell eventually leads to the production of two identical cells. Let's take a closer look at this process. During interphase, the DNA and organelles double and proteins necessary for mitosis are synthesized. Following interphase and the replication of DNA, the cell enters prophase, when the DNA condenses into structures called chromosomes, and the spindle apparatus begins to form in the cytoplasm. The spindle apparatus, a complex of microtubules and proteins, helps to separate the duplicated chromosomes in a later step. Following prophase, the cell enters metaphase, when the membrane surrounding the nucleus dissolves and the duplicated chromosomes migrate to the center of the cell along the metaphase plate with assistance of the spindle. Now the chromosomes can evenly divide and separate during anaphase. Up until this time, the duplicated chromosomes have been joined together at the centromeres and are referred to as sister chromatids. When the sister chromatids separate, each chromosome gradually migrates to opposite ends of the poles of the cell while being pulled by the spindle fibers. At telophase, nuclear membranes form by surrounding each complete set of chromosomes, and the spindle fibers break down. The final division of the cell occurs during cytokinesis, when the cell splits completely in half, forming two cells that are exact duplicates of the original parent cell. The new cells are called daughter cells. This entire process can take as little as 20 minutes in bacterial cells and up to 12 to 24 hours in mammalian cells. The rate of timing of mitosis is controlled by a variety of genes that are turned on or off by chemical signals that are passed between neighboring cells. Meiosis is another form of cell division and is exclusively for producing cells that are used for sexual reproduction. The parent cells of meiosis are called germ cells, and they lead to the production of daughter cells called egg and sperm cells. Meiosis proceeds like mitosis, with DNA replication occurring first during interphase. Then the cycle will go through similarly named phases like mitosis, but it will go through two cycles of division, yielding 4 cells rather than 2. One significant difference between meiosis and mitosis occurs during prophase 1. After the duplicated DNA has condensed into chromosomes, the chromosomes pair with their homologous matches. For example, if an organism has 2 chromosomes, it possesses 1 homologous pair. While paired, the homologous chromosomes will swap sections of DNA, which contributes to the genetic diversity that result from sexual reproduction. This process is called crossing over. During metaphase 1, the nuclear membrane dissolves, and the chromosomes travel to the metaphase plate, where they line up opposite their homologous pairs, rather than singly, as in mitosis. These pairs then migrate to opposite poles of the cell during anaphase 1 and telophase 1 until the cell completely divides during cytokinesis. This concludes the first meiotic phase, and the 2 cells immediately enter the second meiotic phase without any further DNA replication. The second meiotic phase proceeds in sequence just like mitosis. The sister chromatids are separated during anaphase 2, and 4 daughter cells are produced by the end of the second meiotic division. Because of replication of DNA and the 2 sequences of division, the 4 daughter cells all have half the original number of chromosomes as the original parent cell. This is ideal for sexual reproduction, where a sperm cell and an egg cell fuse to form the first cell of an offspring called a zygote. This zygote has a complete set of chromosomes and can proceed through growth and development through the process of mitosis. Now we can compare the two basic processes of cell division. Both processes begin with DNA replication and then a sequence of phases. Daughter cells produced through mitosis are genetically identical to the parent cell and have complete sets of chromosomes. Daughter cells produced from meiosis are genetically different from the parent cell due to the reduction in chromosome number and also the process of crossing over. Meiosis results in the formation of 4 daughter cells, while mitosis creates 2. Thank you for watching this video on mitosis and meiosis. Please visit the Purdue Global Academic Success Center's YouTube channel to watch more videos like this.